Assalamualaikum everyone, this is RPHVR and welcome to the platform of Pharmapedia once again with a new topic, that is mutation. What is mutation? Mutation are changes in any nucleotide sequence of the DNA may occur in the somatic cell. What are the somatic cells? Somatic cells have tendency to not pass to offspring. If the mutation occur in the gametes are the egg and the sperm that have a tendency to pass to the offspring. Are mutation helpful or harmful? Mutation happen regularly. Almost all mutation are neutral. Chemicals and UV radiation are the common causes of mutation. May mutation are repaired by the enzyme, but sometimes it is not repaired by the enzyme and transmitted to the offspring. But if it is transmitted to the offspring, it is helpful or harmful. Some type of the mutation, like the some type of the skin cancers and leukemia result from the somatic mutations, are basically the harmful because it can tendency to transmit the cancers from the parents to the offspring. But some mutations may improve an organism's survival, so it is obviously beneficial to the offspring. Now, the type of the mutations are further two types, that are chromosomal mutation and gene mutation. Chromosomal mutation may involve the chromosome. As the name indicates, chromosomal mutation, this term is self-explanatory: the loss or the gain of part of a chromosome. Now, chromosomal mutation have further five types, that are deletion, inversion, translocation, non-disjunction, and duplication. Now, what does these five types mean? Deletion, due to the breakage, any part of the chromosome or the any piece of a chromosome is lost. As you can see, the pink part is deleted and the chromosomal size become shrink. Now, in the inversion, that means some part of the chromosome is inverted. Chromosome segment break off, segment flip around backward, segment reattach, that means it is inverted. Now, this is a picture, you can see break in the chromosome and it is inverted, flip, and again reattach. Now, in the duplication, as the name indicate, occur when a gene sequence is repeated. Translocation involve two chromosomes that are not homologous. Part of one chromosome is transferred to the another chromosome, that means it is translocated. What does it mean? As you can see in the normal chromosome 20, green and the red part, this is translocated and it is not homologous, that means it is not same. Now, a small part of the red portion is attached to the green part and the green portion attaches to the red part, that means it is translocated and transfer its part, and obviously it is not homologous. And this is the translocation between chromosome 4 and 20. Chromosome 4 in the green color and chromosome 20, that is in the red color. These both are the non-homologous. Now, non-disjunction, failure of chromosome to separate during the meiosis, causes gamete to have too many or too few chromosomes, and this is basically the picture, the process of the meiosis. Meiosis 1 started normally and line up in the middle of the cell. Tetrads are basically line up in the middle of the cell, as you can learn in the ninth class biology. Then one side of the homologous does not separate and meiosis 2 occur normally. All gametes have an abnormal number of the chromosome, either one too many or one too few. So this is basically the non-disjunction. Not all of the gametes are divided equally, and all of the gametes have the abnormal number of the chromosomes because of the improper division. Now, chromosome mutation animation, this is the original chromosome. This is basically the animated picture, so you can learn and understand easily. This is not the actual picture. original chromosome A, B, C, D, E, F, G. In the duplication, you can see the D part is duplicated. In the deletion, you can see the D part is disappear. In the inversion, you can see E and F are inverted. And the inversion, again, you can see the B and the C part. Now the another type of the mutation is gene mutation, that is a second subtype of the mutation. First type is a chromosomal mutation and the second type is gene mutation. Gene mutation is change in the nucleotide sequence of the gene, may only involve a single nucleotide, may be due to the copying error, chemical viruses and the etc. Type of the gene mutation, obviously the same three: substitution, insertion, deletion, and the other two are point mutation and frame shift. Point mutation, change of a single nucleotide, include the deletion, insertion, or substitution of the one nucleotide in the gene, and this is the example of point mutation. Sickle cell disease. Sickle cell disease is caused by the point mutation, and as you can see the normal picture of the RBC, and in the sickle cell disease the RBC become sickle cell, so there is no hemoglobin and there is no oxygen transported to the body. It can lead to the hypoxic condition and it is very lethal condition. Now back to the topic. Sickle cell disease is the result of one nucleotide substitution, which is basically the point mutation occur in the hemoglobin gene. Frameshift mutation. What is the frameshift mutation? Inserting or deleting one or more nucleotide, changing the reading frame, like changing a sentence. Protein build incorrectly. Frameshift mutation are further two type: original, the fat cat ate the VRED, and frameshift E added the fat cat Ted her ewe. That means this is a original sentence. And in the frame shift you can see the cat T is substituted with A. So this is the basically change in the original sentence, and it is the example of frame shift mutation. Amino acid sequence change. Frameshift mutation, here you can see the amino acid sequence, but in the mutated gene sequence the gene sequence is altered. Hope you get the basic concept of the genetics and the mutation. This